On this, the 5th day of November, the Rockefeller University honours you, Suzanne Cory, as the recipient of the 2009 Pearl Meister Greengard Prize, an international award recognising outstanding women in biomedical science. Throughout your remarkable career, you have helped to elucidate fundamental mechanisms of immunity and clarify key events that lead to cancer. As director of the Walter and Eliza Hall Institute, the first woman to hold this post, you inspired scientists worldwide as you guided that distinguished institution into the 21st century. Today, in presenting you with the Pearl Meister Greengard Prize, we recognise and thank you for your extraordinary scientific insights, your numerous contributions to biomedicine, and your continuing leadership in the global community of scientists. Thank you. I think that now you have to read a citation.
Thank you. It is my honor this evening to present Dr. Suzanne Cory the Pearl Meister Greengard Prize, an international award recognizing outstanding women in biomedical science. On behalf of the distinguished jury of the Pearl Meister Greengard Prize and all of us here today, we congratulate you on this award.
We're going to have a few minutes of sort of questions and answer. I'm going to answer a couple of questions, then we're going to go around
You're going to
the
answer
audience.
them, are you?
Did I say answer? I'm going to ask them. I'm going to ask them. I'll answer them too, if you
yes,
like.
Please.
No, yeah. So I think I'd like to ask you, what is the piece of work that you're most proud of that you've done? What's the nicest piece?
I think the Mick mice were really important at the time, and I'm really proud of that. But David Vo's discovery of BCL2 being such a different oncogene opened up an entirely new world, and that was really exciting as well. I can remember those cells. You know, we had them in. He had them in culture. And they were there without cytokines for days, and they just kept shining and looking at you. They didn't divide, but they just kept surviving. We knew that this was something very different. So I find it hard to choose between the two. They're my favorite genes.
And what was it about those two discoveries? Is it that it was a shock for you, just very different, or was it that the approaches you were using were particularly elegant, or what was it about it?
I think it's because they opened up totally new fields of science. I mean, many people were involved in these discoveries. I certainly don't take full credit for them. But after these points in the history of cancer research, whole fields opened up. For example, many other different oncogenes were able to be isolated once it was realized this really was a translocation to the immunoglobulin locus, and you could use these as tags to isolate the genes. So a huge stream of work started from that. But also very importantly, I think our mouse model enabled cancer researchers to study the very early stages of disease, because MYC was not sufficient. Other genetic changes had to happen, but using the mouse as a test tube, you could watch as these changes evolved. Now that was a new concept at the time, and of course that couldn't have happened without the generation of this technology by Palmater and Brinster. So, and then on the BCL2 field has just totally transformed the way we looked at the development of cancer and the kinds of synergistic mutations that have to occur. And again, it's opened up a field that I think is going to generate very powerful new technologies for treating cancer. So both exciting fields, and one led to the other, of course. But I think that if I look back on things, these changes happen because people have developed very new and powerful technologies, and there's a quantum leap jump in what you can do. I mean, sequencing, Fred Sanger, recombinant DNA technology, and the whole genome opened up. Transgenic mouse technology, that opened up a whole new era of biology. So scientists are very limited in what they can do until a new technology comes along and transforms the horizons.
Did you ever manage to have a conversation with Fred? Because he's always so
quiet
and there in his lab.
Absolutely. He was great. I always thought, actually, he was an Australian who was a UK citizen in disguise. He was so nice.
Not quite sure how to take that one.
Well, you can be very formal, can't you? Well, you being the generic British.
The generic British, you mean?
Yes, not you, of course.
Yeah. I think I better move on. I feel a hole opening up in front of me. I'm going on the offensive. I had a look at your website, and I had a feeling that everybody in your lab might be female, or nearly everybody.
They are at the moment. This is the very first time in my life, but actually, by chance, they are at the moment.
It is by chance, because I was going to ask, is this by chance or design? You know?
No, not design at all. I never consider gender, only ability.
Has it changed the way you operate? I mean, you know, compare with a normal sort of 50-50, or is it anything different about it?
Yes.
I'm really not doing well here, am I? A male Brit is really in trouble. Yeah. It's very friendly.
Yes.
Do you think that? Do you have any view? Do women do science differently in your view from men? In a different sort of way? Do they put a different emphasis on things?
I want to answer it a different way. I think that women take longer to develop as scientists because they don't trust themselves enough. They feel, so I think this is very important. This is my own personal observations, no data, so I'm probably totally wrong, but I think that women don't like to move until they feel very confident that they understand everything. And so men are much more likely to jump in the deep end and learn by the doing. I think women, it takes longer for them to develop confidence in themselves, and I think as mentors, one has to be aware of that and keep encouraging and make them realize they actually do know as much as the guy next door, and they're just as able, and they've just got to jump in the deep end, like I did when I went to Cambridge.
Right, I think we should open up now. I think we've got microphones. Have we in all three? So we've got a few minutes. So if anybody's got a question, wave at me. We have one in the very front. Who can get down here quickest? Thank you. Just here.
Hello.
Hello.
Initially, when you were talking, you mentioned the fact that women have a hard time balancing work and family. And that it's a problem because a lot of women are leaving science, choosing to stay at home, maybe with their family. How are you able to raise two daughters?
I had
and
a
having
wonderful
such
partner,
okay.
and we shared a lot. I was talking to Faces, people in Faces this afternoon. I think there were many factors. Of course, it wasn't easy. And I was very fortunate to have Jerry, who not only is a very wonderful human being, but we were working together, so we could always fill in for each other, either for the family or for the lab, and that made a big difference. I think the other thing that we did right was that we didn't have children too early, so we were established. Our names had been established before we took this risky step. At least for me it was risky, it wasn't risky for him. I didn't know how I was going to manage. And the other things were just simple things, geography. I mean, we bought a house close to the lab, and there were good schools and a good creche close to the lab, and so we didn't have to spend huge amounts of time driving around, and so, you know, also lots of practical things helped. And I had a wonderful sister who happened to be a teacher, so she could take my children in the whole school holidays, and I could keep working in the lab. You know, there are lots of factors. Wonderful mother, wonderful sister, wonderful husband. They're the three most important.